Good day, class. My name is Ivan Somwango from the School of Pure and Applied Science, Department of Biological Sciences, and today we're going to cover the unit Environmental Biotechnology. Today we'll cover just two topics, that is bioremediation and phytoremediation. But before we begin with the two topics, we remind ourselves about environmental biotechnology. So environmental biotechnology refers to the application of biological agents, organisms, or their components, along with chemical, physical engineering processes to maintain, protect, or restore the environment. So biological agents are things like microorganisms, plants, and animals, which we use to control environmental situations. Their components could be things like enzymes, antibiotics, bacteriocins, which, for example, bacteriocins can be used as insecticides to control environmental situations. So environmental biotechnology is wide. As we have discussed before, the areas we normally cover in its applications are like bioremediation in waste treatment, like wastewater treatment, in biomining, where we use things like microorganisms to help in mining instead of using smelting or chemicals to release your metals from the ore that you've mined. Then we have biocontrol and biomonitoring, where we use living organisms to monitor changes in the environment. Maybe if you see plants are dying, that could be an indication of some contamination. If you see certain plants growing in certain areas, an indication of maybe a particular chemical or a particular substance or a particular condition within the environment. Biocontrol would be things like the use of bioinsecticides, which are natural, and they're used to control things like pests. So today we are going to cover the topic of bioremediation. And bioremediation, by definition, it refers to the use of living organisms to remove pollutants in the environment. The term bioremediation is quite wide because it talks about living organisms and remedy, that is treatment, and it can apply in different situations. But we generally refer to it when we are talking about microbial remediation of soil. So when we are talking about bioremediation, we will mainly focus on remediation of soil using microorganisms. Other terminologies which we normally use are biotreatment, biorestoration, and bioreclamation. And when we say we are remedying the soil, what are we remedying? We are trying to remove contaminants in the soil, and some of the contaminants are things like chlorinated solvents, hydrocarbons, polychlorobiphenyls, and metals. Usually these contaminants get to our soil as a result of what we call anthropogenic activities, which are human activities such as industrialization, where industries now pour their waste into the environment, and also urbanization, where you have many people in an urban area maybe releasing things like detergents, which would contain some of these contaminants that I've talked about. Then we consider a contaminant to be a contaminant when it is in sufficient amounts in the environment to cause harm to the human beings, either directly or indirectly, or if it is affecting the environment, because in the environment we have ecosystems which we have to balance to ensure that our environment is safe for our use and also for animals and other living organisms. So like I said, bioremediation, we will focus more on microbial bioremediation, and usually the process uses microorganisms that are already available in the soil, or in certain instances where we can add some microorganisms onto the soil to be able to help us to remedy the soil. So therefore, you can either have natural present microorganisms or selectively inoculated, where you have selected a few of them to inoculate onto the soil. And most of the time, the contaminants we target are organic chemicals, but in certain cases, some microorganisms could be effective with inorganic chemicals or substances such as metals and radionuclides. And most of the time, the microorganisms that I'm talking about are not used as single species. We use a consortium of microorganisms to ensure that different environmental conditions will result in different microorganisms being able to remove the contaminant from the environment. We have microorganisms that can survive in high temperatures, some in low temperatures. There are those that prefer acidic conditions, some prefer alkaline conditions. Therefore, the condition which favors the microorganism will now be the one that takes over, and the microorganisms which are favorable at that point will be able to perform the activity. So what we normally do is, in bioremediation, we use the already existing microorganisms which use the contaminant as their normal food. So what we normally do is just increase the number of the microorganisms by optimizing their growth by adding key nutrients such as nitrogen and phosphorus. So the more we have the microorganisms growing, the more they will use up the contaminants, and hence we'll have remedied the environment. And the rate of bioremediation depends on a number of things, like the type of contaminant, the oxygen supply, the moisture, the nutrient supply, temperature, bioavailability. Now, bioavailability is a situation where the contaminant is in the environment, but it is not available to the microorganism. Reason being, it is not in a state that the microorganism can absorb it. Probably the microorganism would prefer the substance to be in solution, but yet it is out of solution, or it is solid. Hence, the microorganism cannot access that contaminant. Hence, we say bioavailability of the contaminant. Is it available to the microorganism? So that could affect the rate of bioremediation. Then concentration of the contaminant, and obviously presence of microbe inhibitors, which would inhibit the growth of the microorganism. The process of bioremediation could take three forms. The microorganisms could use up the contaminants in either three ways. We have mineralization. This is where the microorganism considers the contaminant part of its food, and therefore uses the food and metabolizes the contaminant together with the food, and hence the contaminant is removed and destroyed from the environment. Then we have co-metabolism. This is a situation where the microorganism may use the contaminant, but it not use it as food, but it will metabolize the contaminant. Hence, it will be removed from the environment. Now, in co-metabolism, look at it like the situation where you have, you take medicine with your, as a human being, you take medicine. So you find that you metabolize the medicine, break it down into a simpler component that can be excreted from the body, but it is not part of your normal food. So you don't take medicine as food, but you metabolize it within your system. So therefore, microorganisms could also perform the same. And then finally, the microorganism could also perform immobilization. This is a situation where the contaminant is removed from the environment and stored within the microorganism. Therefore, it is not available to contaminate either human beings or other organisms within the environment. For example, metals are typically absorbed and bioaccumulated inside the organism. Now, what are the challenges that we face with soil bioremediation? One is what I've already talked about, bioavailability. You may have your microorganisms within the contaminant, but they're not able to access the contaminant because maybe the contact is not there, or the form of the contaminant is not accessible to the microorganism. We have biostimulation as a challenge. Remember, we are adding nutrients to the soil so that the microorganism can grow and make use of the contaminant. But in this case, you're not sure whether the nutrients you're adding are actually reaching the microorganisms and stimulating them to grow. Then the last challenge is bioaugmentation. This is a scenario where the microorganisms, probably you have removed them and cultured them in the lab, and then to increase their number, that is, and then when you reintroduce them into the environment, they'll be in a different environment compared to the lab. Therefore, their growth rate could not be as good as they were in the lab. Hence, you may have a challenge in growing those microorganisms. So we say we'll have maybe a problem knowing how they behave in the field. So which are the different methods of bioremediation? Bioremediation can be done in two ways. There is in situ bioremediation and ex situ bioremediation. In situ bioremediation, this is when the soil is treated on-site, while ex situ bioremediation, the soil is treated off-site, meaning the soil is excavated and transported to a treatment plant where it will now be treated and probably be returned. Now the difference is that in situ bioremediation is not an expensive process, but it takes a longer time than the other method, and we are not sure about or certain about the uniformity of the treatment, because remember, you're treating the soil and you're not sure whether the nutrients you're adding are reaching the microorganisms. You're not sure whether the contaminants are being uniformly taken care of or being removed or being made harmless. Therefore, we have a challenge there on the uniformity of the treatment. And examples of in situ bioremediation include things like bioinventing, we are going to look at it, bioaugmentation, and electrokinetics, which we are going to look at. Then ex situ bioremediation, the advantage is that it takes a shorter time. You are certain of the uniformity of the treatment because, as you're going to see, you are able to turn the soil around and ensure that there's movement of nutrients and the movement of the microorganisms to ensure that they take care of all the contaminants. The only challenge is that it is expensive because you have to excavate the soil, you have to transport it to the plant, and also you need equipment to be able to handle that whatever you're dealing with. Therefore, it could be a bit expensive compared to in situ bioremediation. And the methods normally employed include biopiles, composting, land farming, and slurry phase treatment. So let's look at these different types of bioremediation and see how exactly they are done. So the first one is bioinventing, which is an in situ bioremediation. The main idea in this bioremediation process is to increase the amount of oxygen reaching the contaminated zone. In this situation, the extra oxygen will help the microorganisms to grow. It will aid in their microbe growth. However, this particular method of just infusing oxygen into the soil is not very effective, especially in cold countries, because cold temperature retards the activity of the microorganism, hence retarding the growth of the microorganisms. And even if you add your oxygen, then it will not be able to perform as well. Another method is bioaugmentation. Bioaugmentation is what I mentioned earlier a bit, where you take the microorganisms out of the soil. You select the ones you want to improve on, maybe culture them in the lab so that you increase their growth. And in certain instances, you can also engineer them so that they can perform even better, and then you put them back into the soil. So you can collect them from the site and then return them to the site once you have already cultured them in the lab. The idea behind this is also, in the soil, we have so many microorganisms. So you want your microorganism that is very active to the contaminant to be dominating. So you grow it faster, and when you return it to the soil, it will be the one dominating. And when it is dominating, then there's less competition from the other microorganisms for the nutrients that you're going to add. Hence, they'll be more active in removing your contaminant from the environment. And the last in situ bioremediation method is electrokinetics. In this process, we use electricity to move around the nutrients, the water, and also to heat the soil. Remember, heat is good for microorganisms. If it is in their optimum region of performance, then they will perform better if the soil is heated. So we normally insert electrodes. As you can see, we have an anode there and a cathode, and this is the area where you have the soil. And then an electric field. You can see there, those lines, an electric field is being passed across. And you find that biomolecules, meaning your water, your nutrients, they normally have charges. Some of them would have charge, especially when they're in solution. Therefore, depending on the charge on the biomolecule, then it will move towards the different electrodes. The idea is to move the nutrients around so that different microorganisms can be able to access the nutrients as they pass by. And the nutrients now are not static. Then we have electrolyte pumps, which now actually pump the nutrients into the soil, while the electrodes now create that field where now the nutrients will be able to move around. This way you are ensured of the growth of the microorganism, and then it will take care of the contaminants. Now, ex situ bioremediation, like we said earlier, is where you do the actions off-site. You pick the soil, you excavate it, you transport it to a treatment plant. One method is biopile treatment, where, as you can see in the diagram there, this is a pile of soil, okay, or which has been collected from a site. Now the treatment area has two additions, where you have the air injections. We have now aeration system where you're injecting or infusing air into the soil. The idea is to put in oxygen to the soil to enhance their growth. And then you also have an area where you're infusing nutrients, moisture into the soil, again for purposes of growing the microorganism. Once they're grown, they will take care of the contaminants by themselves. Then the area is also covered by, you can say, a film, okay, plastic film, okay, which now prevents whatever is leaching. You can see leachate collection treatment. Whatever is leaching, remember leaching is when the minerals or even the contaminant mixes with some water and then percolates down the pile and can go into the soil in the region where the treatment is being done. So this plastic covering prevents the movement of leachates down to the soil, hence the area is controlled. So that is biopile treatment, where you pile the soil and then you treat it as when it is in a pile. Then we also have land farming, which is almost similar to the biopile treatment. In land farming, as you can see, the arrangement is the same. You have the plastic liner, okay, this one going round, and then you have the soil. But in this situation, the soil is not in a pile, it is flat, and the idea is to again add nutrients for the microorganisms to grow. And you have, I don't know whether you can see on camera, my small tractor there, which is trying to till the soil. The idea is to aerate the soil, okay? It is mixing the soil up so that air can get into the soil. In biopile, we had some tubes running into the soil which would infuse the air into the soil. So the difference here is you use a mechanical or a tractor or a mechanical system to move the soil around. So the rate of degradation is controlled by nutrients which are added externally. And lastly, there's another one more, but in slurry phase treatment, this one is a bit different, where the soil is put inside a bioreactor. Now, a bioreactor is a container which we normally use in biotechnology to control the environment for a microorganism so that we can optimize the growth of that microorganism. So we control things like the air, meaning oxygen, whether we want oxygen or not. We control the pH by either adding alkaline solution or acid solution. We control many other things within the bioreactor, temperature and many other things. So we control the environment so that we have optimum growth of the microorganism. So in slurry phase treatment, the difference is that it's not in the field. You use a bioreactor, which is a container. And you control the nutrients, and you can add things like oxygen, whether the process is aerobic, and maybe do not add oxygen when the process is anaerobic. And lastly, under bioremediation, we have the process of composting. So composting simply means piling up the soil and allowing the microorganisms to convert the contaminants to less harmful forms, or even to remove them completely. So we can either have aerated piles, you can have tunnel systems, or you can have rotating drums, okay, where you pile it in a drum and the drum is rotated. The idea behind rotating is to agitate the mixture so that it mixes up and the microorganisms can have access to the contaminant. So many designs are possible. The ones I've listed here are like aerated static pile composting, which you have piles which are of the compost, which are aerated by blowers or vacuum pumps. Again, you add the air to encourage the growth of the microorganisms. Then we have mechanically agitated in-vessel composting, where the soil is in a vessel. The vessel is there to both agitate the soil, shake it around, and also to aerate the soil for the reasons which we have mentioned earlier, so that the microorganisms grow and they're able to use up the contaminants. Then lastly, we have windrow composting, where the compost is placed in long piles known as windrows, and they are periodically mixed using mobile equipments like tractors or excavators, so that you mix them around and allow the contaminants to use, the microorganisms to use up the contaminants. I have a diagram of an example of windrow composting. As you can see, they have your piles there, and they can be mixed around over time. So this one now allows the microorganisms to do their work slowly. So finally, we'll look at the other topic of phytoremediation. Phytoremediation is also a system of using up contaminants in the soil and also in water. But in phytoremediation, the difference, we don't use microorganisms, but we use plants. So phytoremediation uses plants and their associate microbes to destroy, remove, detoxify environmental contaminants in soil or water. So we can have four methods. You can degrade the contaminant, you can accumulate the contaminant, you can dissipate the contaminant, where you convert it to a volatile form and release it in the atmosphere, or you can immobilize the contaminant. As you can see, we have different techniques. We have rhizodegradation, phytodegradation, that is for degradation of the contaminant. In my diagram here, I hope you'll be able to see them. There's phytodegradation, which happens on the upper part of the plant. There's rhizodegradation, which happens in the root area of the plant, where the contaminant is degraded either in the roots or on the upper part of the plant. Then we have accumulation, where the contaminant can be accumulated on the top part of the plant, which we call phytoextraction, or on the roots, which we call rhizofiltration. Then you can also take up the contaminants through the roots and convert them to a volatile form and release them through the leaves through what we call evapotranspiration, and then the volatile will be released from the soil. The contaminant will be released from the soil into the atmosphere, but by the time it gets to the atmosphere, it will not be a contaminant, it will be converted into another form. Then you have phytostabilization, where the contaminant is immobilized around the roots, and hydraulic control, which we are going to see what it involves. So if we quickly go through, phytodegradation, like I said, is degradation, but the key difference is that here, the enzymes that are used to degrade the contaminant are produced by the plant, and they're not produced by the microorganisms. All plants, or most plants, usually where they are growing, there are certain microorganisms which associate with them through what we call symbiotic relationships. They help each other out in their survival. So in phytodegradation, these microorganisms that grow next to the plant do not assist in their degradation. An example is the poplar trees, which have been used successfully in degrading contaminants where they have been grown. Then we have rhizodegradation, which occurs in the root zone. In this case, the enzymes that degrade the contaminant could either come from the plant itself or from the microorganisms that are growing next to the plant or inside the plant. And most of the time, since they excrete the enzymes, the contaminant is destroyed outside the plant. And this process, you don't need to harvest the plant in order to remove the contaminant, because the contaminant has not entered the plant. It is it's still in the soil, but it has been made harmless through enzymes that have been excreted into the soil. Then we have phytoextraction. Again, contaminant is removed, and broken down within the plant, but absorption is done in the plant tissues, and phytoextraction is above the ground, and phytofiltration or rhizofiltration is in the root systems. Okay, so then the other one I talked about earlier, phytovolatilization, where the contaminant is taken up and volatilized. The contaminant, as it is taken up by the plant, it is converted to a non-harmful form, and then through the evapotranspiration system of the plant, the contaminant which has now become less toxic, is now released through the leaves of the plant and released as a gas. An example I've given there is toxic mercury, which is converted to elemental mercury, which is now less harmless. Toxic selenium, which is converted to less toxic dimethyl selenide gas, which now is released from the plant. So it is taken up by the roots, metabolized within the plant, and then dissipated into the environment in a less harmful way by the leaves of the plant. Then we have phytostabilization, where it's an immobilization technique where the substance or the contaminant is immobilized. It may be metabolized or not. But it is stored within the plant. And this one requires plants that can be able to withstand having the contaminant within the particular plant. So we need some special plants to do this. Another thing they help with, the plants reduce wind, water, and water erosion, which now again blocks the movement of the contaminant through dust, through runoffs, and through leaching. An example is when we finish up with mines, we normally plant vegetation around the mine area so that now the contaminants cannot move. There are various advantages and disadvantages to this process, as you have seen. Then we have the hydraulic control, where most this is normally done to control the groundwater. So it controls the flow of groundwater through the uptake of large volumes of water, prevents leaching, and it induces upward flow of the water table. Now this is a control system where we ensure that the contaminants do not leach down into the groundwater and contaminate the groundwater. Then plants can also help with phytoremediation of air. This is now different from the other processes we talked about. They can absorb different gases like CO2. We all know that photosynthesis requires carbon dioxide. They also are very efficient sinks of polyaromated hydrocarbons, okay, which are compounds which are very difficult to biodegrade. Therefore, as a sink, they take up those compounds and store them. Therefore, we don't have to inhale them when they are in the air. They also take up suspended particles and aerosols, which cause things like asthmatic attacks and all that, by trapping them, either in the leaves or in the stems. But when they are trapped, these contaminants are not now available in the air to cause problems. They also can bioaccumulate pollutants, like the way I said earlier. They store their pollutants in their cell tissues. Therefore, the pollutants are not available to cause contamination. Then they can also be good bioindicators. Like I had said earlier at the beginning of the lecture, that presence of certain plants would indicate something. Absence of certain plants in particular areas could be an indication of something. If, for example, you see a plant is flourishing very well, it gives you an indication of the level of nutrients within that particular soil. And we have other different types of indicators which can even tell us the air quality and such other things that are necessary for us to know whether we have pollution in the air or not. Finally, we have different applications of the phytoremediation techniques that I have just talked about. We have what you call vegetated cap, which I had already talked about, where you plant vegetation over a contaminant, which now blocks its movement. We have the buffer strips, where you cause a buffer between the contaminant and the other areas where you don't want contamination to happen. Sometimes we call them riparian corridors, especially over maybe a waterway and some other contaminants maybe coming from industry. Therefore, the contaminant will have to flow through the vegetation before they get to the water. In the process, the plants will be able to pick up the contaminants and they will not be able to get access to the water. And then finally, like I've mentioned before, environmental monitoring. Plants can be used for phyto investigation or for environmental monitoring, where we monitor air pollution by analyzing the tissues of the plant. There we can be able, if the plant is bioaccumulating, we can be able to check the levels of certain contaminants in the environment by just analyzing the tissues of the plant. We can also have some plant species which give us clues to things like the presence or the depth of groundwater. Like I'd said, the presence of certain plants is an indication of something. Like, for example, the presence of acacia trees would indicate that that area is either arid or semi-arid. If you see many acacia trees, it tells you water is a bit scarce in that particular area. So those are the two methods I wanted us to learn today: bioremediation and phytoremediation, where we use plants and microorganisms to remedy contaminants in soil or in groundwater, or even in certain cases in surface water, so long as it is static and the actions of the plant or the microorganism can be allowed to work over some time. So with that, I want to thank you for your time and ask you to stay safe. Thank you.
These televised lectures supplement our robust online learning going on our MKU online platform. You can view more on our televised lectures via our online platform. We are in a digital era, and Mount Kenya University knows this. The following are the steps to follow so as to complete your online application. Download the application form from the website www.mku.ac.ke. Attach copies of your academic certificates and ID. Pay the application fees via M-Pesa pay bill number 270988. Your ID is the account number. 2,000 shillings is the charge for a postgraduate. You can also deposit in the bank accounts provided on the website. Then email all the above to apply@mku.ac.ke.